For our next topic, I'm really happy to introduce Dr. Timothy Yu as our next keynote speaker. Dr. Yu is a neurogeneticist at Boston Children's Hospital and Harvard Medical School, and his research focuses on human genetics, neuroscience, and molecular medicine. So his keynote lecture is going to be on hyperpersonalized therapies for the long tail of genetic disease. So welcome.
Thank you very much for that kind introduction. It's a real pleasure to be here with all of you and to share with you our work. I'd like to suggest that our work really builds upon the successes that you've all been discussing over the 5-year history of the Solve-RD program, giving us an opportunity to diagnose patients and then take the next step to think about ways in which a large body of these diagnosed patients can be used to find opportunities to intervene and to help and advance treatments for many of these conditions. I realize that I'll need this, so let's not get too far away from it. And motivated by, like many of you in this room, I began my career in human genetics some 15 years ago with a real focus on trying to apply tools for next generation sequencing to help discover new genes and then to diagnose patients. And I think the reports that we've seen in this Congress have shown how remarkably far the progress has taken us. This type of sequencing, advanced reanalysis, functional validation is hugely shortening the diagnostic odyssey for patients with genetic conditions. And that increasingly encourages us to focus on the next step, which is to tackle this challenge, which is that treatments for these conditions remain few and far between. Now, luckily, however, I think that this is beginning to change, and I hope that my talk will convince you of this. What we are beginning to see is the fulfillment of a promise first offered some 25 years ago with some of the first gene replacement therapy publications. We are now seeing in 2023 a growing wave of such genetically targeted therapies that have expanded beyond traditional viral gene replacement therapy into many other modalities like mRNA therapies, antisense oligonucleotides, CRISPR editing, siRNAs, and so forth. And in particular, I'm going to be focusing on some work that we've been doing recently with antisense oligonucleotides. Antisense oligonucleotides, for those of you who may be familiar but not necessarily work with these on a daily basis, are short snippets of synthetic RNA or DNA-like molecules. They have been chemically modified for good pharmacologic properties, but they're remarkably simple to manufacture because of their modularity. It turns out for clinical and biological luck that they're very easy to deliver to the body, especially to the central nervous system. If you give them intrathecally, they can distribute broadly throughout the brain and spinal cord, and they don't require adjuvants, they don't require vectors, lipid nanoparticles. A simple delivery of naked antisense oligonucleotide in saline is sufficient to give excellent biodistribution. And once distributed within their targets, they can be designed in such a way to bind to specific mRNAs or pre-mRNAs to alter patterns of gene splicing or to target genes for destruction. The canonical example of an ASO, which folks are well familiar with, is an antisense oligonucleotide for spinal muscular atrophy, nusinersen or Spinraza, which was approved in 2018. And the sequence for this drug is shown up here. It's an 18 nucleotide synthetic RNA. It's given by spinal tap 3 times per year. And I think that it's had transformative clinical benefits to many, many patients, over 20,000 individuals so far. And it's such a success, I think it requires very little explanation. An interesting facet of this disease, however, which I'd like to point out, is that it is a mutation-specific therapy. It acts on the SMN2 gene, a evolutionarily duplicated copy of the SMN1 gene, which is present in each of us. And during that evolutionary duplication of the gene, it acquired a point mutation that we all carry that leads to exon skipping of exon 7. And the purpose of the nusinersen drug is to target that common mutation in all of us and to restore inclusion of exon 7. The point that I'd like to make is that this is actually a very unusual situation. That by doing this, by targeting this acquired mutation that happens to be found in all of us, this is taking advantage of an evolutionary loophole that is by and far the exception and not the rule. That requires us to think, if we are to use technologies like ASOS in the nusinersen class, what do we do if the mutation that we're targeting is only found in a small number of individuals, as is most commonly the situation, or even just a single patient? We began thinking about this problem because we were very fortunate to begin working with a very special case that of a young girl with Batten disease that we published now 3 years ago. And this is published work that I will refer to just for framing, but we demonstrated that we were, under very particular circumstances, able to use a nusinersen-like strategy to develop a splice modulating ASO for a young child with Batten disease. This was a 6-year-old girl who had been diagnosed with Batten disease after 3 years of progressive symptoms, after a diagnostic odyssey familiar to everybody in this room. She had been diagnosed with CLN7 Batten disease, an ultra-rare condition that is predicted to be fatal by age 11. And her diagnosis was made initially on a clinical basis with a wrinkle, which is probably also familiar to many of you in this room, which is that she had only half of a molecular diagnosis. Her conventional clinical sequencing had only found one heterozygous mutation in a recessive gene, the CLN7 or MFSD8 gene, and the second mutation was not to be found. And it was through efforts like described here, of reanalysis, whole genome sequencing, and then reanalysis, that we uncovered the solution to this puzzle, which was that whole genome sequencing showed through IGV browser gazing, like was demonstrated yesterday, a cluster of unusual deep intronic reads that led us to recognize the presence of abnormally paired reads in intron 6 of the CLN7 gene. And these abnormally paired reads, their particular pattern, which I won't go into the details because, as I mentioned, it's already published, illustrated the insertion of a mobile DNA element, a deep intronic insertion of an SVA retrotransposon, which had interposed itself between exon 6 and exon 7. And the functional impact of this, which we demonstrated by RNA sequencing and other methods, was to create a new splice site that disrupted proper gene assembly. And that was our diagnostic odyssey to solve a very interesting Mendelian puzzle that led to completion of the diagnosis for this previously half-diagnosed case. It was also, however, a very fortunate observation that led us down an unexpected path, which is it gave us a therapeutic opportunity to intervene here with a customized antisense oligonucleotide that we reasoned could be used to block the abnormal splice event and restore normal gene splicing. Just conceptually, how this process went was to employ patient-customized antisense oligonucleotide design, targeting the novel splice junction, to test these in patient cell lines, initially fibroblasts and then iPS cells, and to screen a series of drug compounds looking for one circled in green that could reverse the normal pattern of abnormal splicing found in our individual case. And we were able to quickly find a drug candidate that was able to boost the amount of normal splicing by 3 to 5 times and enlist in a widened team of FDA industry and regulatory advisors, a streamlined manufacturing and animal safety plan that allowed us to develop this drug and begin treating her with it in 1 year's time. And as reported in that paper now 3 years ago, we had really more success early in this trial than we expected from such an early and first effort. We saw seizures reduce remarkably over the first 6 months of treatment. Her family reported significant quality of life improvements in her responsiveness, in her muscle tone, in her ability to walk. We were very, very fortunate to see this remarkable clinical response early in the trial. Now, as some of you may know, though, in year 3 of the treatment, it also became apparent that while our treatment appeared to be engaging and helping the patient, that it ultimately was not sufficient to reverse the significant 3-4 years of neurologic decline that she had begun to suffer before we met her, and she eventually passed away. But her family's gift of a research autopsy allowed us to demonstrate that in tissues from her brain and spinal cord, we were able to see rescue of normal CLN7 splicing up to 80% of the total measured in brain and spinal cord in a tissue distribution that seemed to mimic the expected biodistribution of our drug, peaking at the injection site in the lower spine, but then also extending to significant rescue in the brain and cerebellum as well. This first case has led to the introduction of this idea and a lot of excitement around this idea of programmable medicines. That with drug backbones like nusinersen and drug mimics like melicin, which use the exact same chemistry, the same delivery method, but a different sequence and a different target, we might be able to flexibly repurpose a platform to accelerate the design of even patient-customized medicines for a wide variety of our patients. And it's the basis for the further examples that I'll share with you, 2 more examples from our work. In the second example, following the melicin case, we encountered a young child who had an abnormal newborn screen. At birth, she was determined to have a low T-cell receptor count, initially concerning for severe combined immunodeficiency. It turned out that she did not have SCID, but she had a condition called Ataxia Telangiectasia. This is a condition associated with progressive cerebellar neurodegeneration, which has unfortunately no treatments. These children suffer progressive neurologic incoordination, and it is unfortunately fatal by young adulthood. And it's caused by mutations in a gene called ATM, which is a critical mediator of the cellular DNA damage response. One of her ATM mutations, however, created a new splice site, which looked like this. This was a C to T point mutation in exon 53. And it created a new splice donor site, which allowed us to play the same trick that we had in the melicin case. We were able to design a series of antisense oligonucleotides that inhibited the usage of this new exon 53 splice site. It rescued the truncation of exon 53 that the mutation otherwise would cause. And in patient cell lines, we were able to demonstrate this rescue of proper splicing in a dose-dependent fashion. We were able to rescue the DNA damage response after irradiation through downstream effectors like phospho-P53 and phospho-Cap1. And we were able to conduct manufacturing and safety studies on this experimental basis to initiate an experimental trial for our patient by age 2. This work here done by a large team in my laboratory in collaboration with the AT Children's Project is the start of an ongoing trial, which is now 3 years into execution. We're happy to say that the drug has been well tolerated for the last 3 years. It is still, unfortunately, too soon to declare success, as the natural history of this condition actually predicts that in the early stages of disease, from 0 to 5, there is a period of relative stability. Our patient just turned 6, and our treatment window has been within this green box right here. And it is in this next phase from age 6 to age 10 where patients typically decline. So we are still watching this readout very, very carefully to try to determine the ultimate success. But with safety as the primary indication for a child in whom we were intervening very early, really before meaningful symptoms had set in, I think that the tolerability of this is our first small success. We have some reasons for optimism that give us the promise that we hope to have a positive readout in the next few years. We have been working with a number of groups, including Anupam Gupta at MGH, who is piloting the use of wrist and ankle biometric sensors to measure longitudinal recordings of motor velocity in the limbs. And Dr. Gupta in his laboratory has shown that this appears to be an effective discriminator between controls in gray and AT patients at a variety of ages in red, where the peak velocity is consistently lower in children with AT and children and adults with AT. Our patient is the blue dot shown there, which illustrates that there's significant heterogeneity within the AT population, but at least our patient is in that range of overlap between controls and AT patients. So we hope that with time, we'll be able to begin to see separation of these curves. I will point out that this N of 1 is not a true N of 1. A second patient revealed himself a year and a half after starting our first patient. This was a patient identified in Europe, and I'm happy to say that this patient, whom you'll hear about later from home, is now receiving experimental treatment close to his home at the University of Tübingen in a trial that we are doing in collaboration with Mathis Sinopsik, Rebecca Schul, and others. And in addition, in collaboration with neurologist Banu Anlar in Turkey, there have been 5 additional patients with this specific AT mutation, which seems to be recurrent in Turkey at Hacettepe University. And we are working hard with them to try and expand the trial as well. Example 3. This is an example of FLV-CR1 associated blindness. This is a young child who actually presented to us from the very same clinician who referred our Batten patient to us. Dr. Austin Larson at the University of Colorado had identified and followed since birth a young child who presented with congenital insensitivity to pain. She had multiple self-injuries as a young infant because of her inability to sense pain and protect herself, multiple digital infections. She also had progressive tunnel vision, loss of night vision, and findings of retinitis pigmentosa on ophthalmologic exam. And her clinical and genetic workup suggested a diagnosis of posterior column ataxia and retinitis pigmentosa, a rare condition discovered by Deborah Chiabrando at the University of Turin that causes degeneration of retinal neurons as well as dorsal root ganglia. And there are, as an ultra-rare condition, perhaps 20 or 30 patients described with this condition, and there are no treatments for it. Clinical testing by Dr. Larson had shown a pathogenic point mutation, a missense mutation, tyrosine 398 to cysteine. But as a recessive condition, this was also a half diagnosis where the second mutation was missing. Well, it turned out that whole genome sequencing again revealed the answer. We found by whole genome sequencing, but there was, again, like the Batten case, an unusual collection of chimeric reads deep within an intron 8 of FLV-CR1. These are the junctions of those unusual reads delineating a breakpoint at which there had been insertion of another mobile DNA element, this time a pseudogene that was mobilized by an SVA transposon, but not an SVA transposon itself. This pseudogene insertion had the impact that our Batten patient's insertion did. It created a new splice site that led to a stop codon 6 codons after the insertion site. And using the same methods that I've already described, Bo Zhao and my group was able to restore FLV-CR1 expression in patient cell lines using a splice blocking ASO designed to this region here. Family discussions led by Emily McCourt, who has been this child's ophthalmologist since birth, focused on the clinical risk benefit for potential intervention here. This child is now 6. She has a sensory neuropathy, which is unfortunately likely to be irreversible, but she's managed to protect herself and learn self-care such that that is actually a clinically manageable neurologic symptom. However, she relies entirely on her vision to navigate the world. And her vision loss there is progressive and her fields of view are restricting and clearly impacting her quality of life. This is her and her family's main concern. Considering the fact that intravitreal ASO dosing through the efforts of groups like ProQR has now been established as a well-tolerated route, we considered whether or not ASO treatment could slow vision loss. There are some cataracts that are associated with long-term ASO dosing at high levels, but considering the clinical impacts of those and that there are readily available surgical interventions to relieve those, might those be reasonable or manageable? And other considerations, could we contemplate a study design where we treat one eye first, use the contralateral eye as the control, and also manage both scientific rigor as well as safety at the same time? And I'm happy to say that this project, which we took on in collaboration with NLORM, was given the green light by the family about a year ago. And the IND filing for this happened just yesterday. So this case is now in front of the FDA for review, and we hope to begin this in the next few months. These cases and other cases from colleagues at Columbia, University of Massachusetts, John Watts, Neil Schneider, and others from NLORM have served as the basis for initial draft guidances for this type of work from the FDA. In 2021, the FDA released a series of draft recommendations for how these types of filings should be submitted to them, for what safety requirements, manufacturing requirements, clinical design considerations should be submitted for their review. And they granted many of the concessions that they had negotiated with us in our initial cases: accelerated review timelines, streamlined requirements for manufacturing and toxicology, for instance, the use of single species for testing as opposed to the usual 2 species requirement, and specifically for situations that involved orphan conditions that were seriously debilitating or life-threatening. And that opens the door really for exploration of this space. The critical question, though, is can these experiences scale? I've given you 3 examples, but can we find more such cases? And can we actually prosecute them? Well, to address just the first of those, how many cases like this exist? I'd like to share with you some work that we've been doing with AT as an example. And to explain this, consider that I've shown you in our first case, in the first case, if we consider nusinersen the first example of a successful mutation-specific therapy, that we have an ASO targeting an intronic point mutation causing exon skipping. Consider that in our Batten case, that we have an ASO targeting an intronic mutation, creating a poisoned exon. It creates a new splice site as opposed to, it creates exon inclusion, not exon skipping. Consider that in the AT case, I've shared with you an example of an exonic point mutation causing the creation of a new splice site, leading to exon truncation. And finally, I've shared with you in our ocular case, another example of an intronic insertion creating a new splice site. We've gone through now and taken these as examples and many, many others, dozens of other examples, to try to create a first taxonomy for these types of treatable mutations, to systematize their discovery. And we've described a number of scenarios by which these treatment opportunities can be discovered, and then gone looking for them prospectively in a cohort of patients with AT. It turned out that the AT Children's Project had previously assembled a registry that recruited 235 cases of AT, well clinically characterized, excellent diagnoses, excellent diagnostic specificity, and performed whole genome sequencing in those. And working with their data set, we applied comprehensive variant calling for the full spectrum of not just point mutations, but CNVs, structural variants, mobile DNA elements, and so forth. We used algorithms for coding and non-coding splice impact prediction, and then applied our predictive taxonomy to plus experimental validation using patient cell lines, RT-PCR, RNA-seq and engineered mini-gene assays when we couldn't get a hold of primary cell lines. And what did we find? Well, first, unsurprising to many of you, whole genome sequencing was by far much more effective than whole exome sequencing in achieving a complete diagnosis in this set. We were able to find the two-hit molecular explanation for 99% of the cases in our cohort, compared to 88% for exome-based measures. But really what we did this for was to understand the therapeutic yield. And what we found was that 15% of this cohort, 15% of these individuals, 36 out of 235, had at least one variant that we predicted to be potentially amenable to an ASO therapy. Importantly, 70% of these cases involved treatable variants that were deep in introns, so that they could only be discovered by whole genome sequencing. Traditional, conventional clinical sequencing in most of the US would have missed these. And taking these estimates, we also looked at another recessive condition to see if they might be replicated in a separate cohort. And looking at published cohorts of patients with Stargardt disease, we found between 9 and 21% of individuals appeared to have an amenable variant according to these criteria. And this suggests that this estimate may apply to other recessive conditions as well. How many ASOs are needed to treat these 36 cases? Well, this is the distribution of those variants, how often they recurred within the data set of 235 individuals. I'll point out that there was one very recurrent mutation found in 13 patients, a deep intronic point mutation, which is the cause of 18% of cases in the UK. And that kicked off a really lovely collaboration with Rita Horvath and her team in Cambridge to understand patients with this particular recurrent variant. There's a clinic dedicated to many of these patients there. There were also additional recurrent variants, but really by and large, many of them had private variants. Running the numbers, one ASO for the recurrent UK variant could treat 1/3 of the cohort. 4 ASOs could treat a little more than half the cohort. Pointing out, however, at the same time, those are pretty optimistic numbers. Treating the entire cohort requires 17 of these. So we'll have to get very, very good at these to be able to develop portfolios like this at scale. So to conclude, just my very last small section is to point out where this is going. And clearly through the efforts of Solve-RD and other large-scale whole genome sequencing clinical application efforts, we will find more and more of these variants more and more frequently. And these opportunities are going to be recognized. The question is, what do we need to do to prepare for them? How do we streamline the development of these investigational interventional opportunities? How do we establish safety and efficacy? How do we manage regulation and even consider things like reimbursement and sustainability? These are big questions that I don't pretend to have the answer, certainly not in this talk. But just to point out that they're very important questions to try to come up with answers for, because the great majority of the diseases that we diagnose in patients using genetic methods are too small for commercial interest. And so if we can aggregate our experiences across these types of efforts, we can develop a new orthogonal way to treat patients, not by their gene name, but perhaps by their mutational impact. This work has been done and is being done in conjunction with a really remarkably growing ecosystem of investigators at multiple sites. I'll point out many overarching groups like the FDA, the Oligonucleotide Therapeutic Society, the N-Lorem Foundation, many individual academic institutions, and a series of coalitions and collaborations, the N of 1 Collaborative, IRD-IRC, One Mutation, One Medicine. I'll point out for this audience especially that there's a very robust set of initiatives launching here in Europe, championed by 1M1M, and I refer you to these two papers that describe some of the emerging opportunities here on the continent. and our own global initiative to launch a coalition of academics, nonprofits, and companies who may be interested in this space under the N of One Collaborative, with the real goal of developing best practices, sharing data, and trying to democratize access to this very early experimental but exciting opportunity. And I refer you to our website and work groups if any of you might be interested in learning more and considering contributing your efforts. We would be really welcome to see any of you join. Thank you very much. I've tried to acknowledge the folks who've done this work as I've gone along, and thank you for allowing me to go a little bit over.
Thank you very much for this excellent talk. It's really exciting to hear about the progress towards the future of genomics and also the possible opportunities for therapies. So let's take some questions from the room.
Yes, one first question. The relationship between timely treatment, possibility of time, and approval of FDA for customized treatment. And this is...
Yeah, thank you. That's a really good point to highlight. And arguably the types of conditions that I showed you in the Batten patient, we arrived too late. We always wonder what would have happened if we had met our patient 3 years earlier, if we could have intervened earlier at the point in her condition. In our AT patient, we were very fortunate to have detected this at birth and begun preparing. And it took 18 months from that point forward to advance it. And in our ocular patient, we had the benefit of time, a slowly progressive condition that had an initial wave of sensory neuropathy, but then the second wave of vision loss that was much more protracted. The FDA has been very responsive in terms of timeliness, and they have imposed manufacturing and toxicology requirements that at a maximum will require some 4 to 6 months of prep work to actually execute. That's for GMP manufacturing and for preliminary safety studies. But I think that that's a very generous allowance compared to the normal 12 months, 18 months that you might get if it were a more traditional trial. I think that gives us some window. Still though, earlier diagnosis is still critical as always.
That was stunning. Thanks so much. I have a little bit of a conceptual question. So while I fully agree that you showed very nice examples where it's really this N is one, this very specific mutation, there may be scenarios where either mutations cluster, for example, for gain of function or deep intronic splice sites, where you could envision that you just design it for an allele. Just you take a 50% SNP and just by random, you can almost treat anyone with that. You just need to see by phasing, and the long reads obviously enable us to see in which phase the mutated allele is, and then you can block that for almost everyone, and you can start designing that up front before even knowing all the details.
Yes, that's an excellent point. And allele-specific therapies are an example of a mutation-specific therapy that I honestly deliberately avoided because it does introduce that extra complication. From my personal stance, that N of 1 has been a very useful name to put a name to this field and to name a name for this type of clinical argument. But to me, the actual critical number is simply whatever the number is required to get a company involved. And if a company and a commercial effort can be marshaled, then perhaps that is a useful practical handle. For orphan conditions that fall below that, for which, again, the condition is sufficiently serious, I think this approach ought to be considered. I think there's a strong ethical argument to advance that. And I think that those fall in that category too.
Hi, Timothy. Was really great and exciting. And I have 3 points. One, doesn't it trigger that we have to have worldwide registries for the different diseases, and how do we manage? Second point, what you mentioned with the ATM story, is how do we accelerate biomarker development, because we have to see whether an ASO works, or works efficiently despite the phenotype is developing slowly. And the third one is probably the most tricky one. You raised already the point that more or less we have to move this despite maybe ethical concerns or whatever, into newborn screening. Now, let's put aside that at least for Germany alone, we would have to sequence whole genome. I think you made it clear, exome panels will not work because of the splice sites and so on. We have to have the genomes. We do not really have a disease sort of selection, so we have to do the genomes. For Germany, it would mean 600,000 newborns per year in time to be sequenced, analyzed, reports to write, and so on. Let's not talk about this one, but if we push the society this way, we would immediately, and coming with your side of the discussion, developing therapies. There would be a huge demand overnight or expectation of patients. And not, a single one. We are talking about hundreds of thousands overnight, basically. And I know that the capacity of companies is pretty good, actually. So in terms of ASO, so they have proven with COVID, they have proven with cancer and so on. They have proven that they can do very tiny ones efficiently, overnight, basically, things. But how do we, from the clinical side, medical side, study side, and so on, how do we get it organized in a worldwide collaboration to have sort of these expert groups selected across the world and making this available to the patients? Sorry.
Those are fantastic questions, Olaf, and I will now answer them in the next 30 seconds. No. Many of them will require constant, continued conversations. So to start, worldwide registries, these patients are so few and far between, and they can crop up anywhere, that absolutely, that international cooperation is the only way to do meaningful science. As our AT example showed, our first patient appeared in the United States, the second patient appeared in Germany, the next patients are in Turkey. And each of these jurisdictions, we've had to reinvent the regulatory mechanisms and the logistical, find the collaborators, and to set up the system for allowing these types of novel approaches to take place. And that's work that cannot be done 100 times in 100 different countries. So there needs to be a unified approach. And I think that's very clear how to actually do it. I think that 1M is doing fantastic work to try and consolidate something under the European Union in the United States, and through the N-of-1 collaborative, we hope to also bring that to other countries as well. It will just take policy effort and databases to allow this type of collaboration. The biomarker question is critical, because in order to develop an evidence base that actually shows that this works, before we talk about setting expectations for hundreds of thousands of infants worldwide, we have to show that it works. And at this point, there have been only a handful of N-of-1 interventions so far. I used to go around and tell folks that I think we need about a dozen of these. And our success or failure in the first dozen will be critical for setting the expectations of families as we begin to prepare for the large scale up. And I don't know what the success rate will be in the first dozen. We've already had some setbacks in the field with adverse events too, some of which are of our own. And I think that if we choose our cases appropriately and find out what percentage of those dozen cases show clinical impact, what percentage might show some safety concerns, that will help us figure out how quickly to communicate and message this expectation to families for that next newborn screening phase. So thanks. I won't try to tackle the third one, except maybe in the panel afterwards, but I think that we'd love to talk more about that.
Can I?
Yes, we will take the two questions that we have here and then one online.
I'll try and have a short question.
Thank you.
All right. So Tim, thanks very much. We remember your presentation at ASHG a few years ago and how that sort of a wave went through the community, which of course is not because you invented a technology, but because your work showed with the Vittarello family that this actually, we can do this. It's like, and that's the sense that sort of permeates, and we felt the same thing today. So thanks very much. You also said you don't have all the answers. Well, yeah. But do you have an, you must see, a vision of how this, in an ideal world, we're not, that's not where we are, but, how would you like this to work in the future? And as sort of, as an element of that, there's a lot of discussion on whether this should be, industry driven, or whether governments, or, the United Nations, should we all get involved at that level? Or do you think it can be done from, the way usually therapies are developed?
Thank you, Han. I think in my title, I've been using the phrase interventional genomics recently. And I have chosen that phrase deliberately because I think it does encapsulate what I hope could be a vision for how this proceeds. And I think what we're seeing, what I hope to express back in 2018 at that meeting with you at ASHG, is that we are at a time when the tools of industry have become sufficiently de-risked that we can contemplate them now being navigated still in partnership with industry, but by responsible professional geneticists and genomicists and interventional physicians in this space. And I think that a hybrid model where we think about these not necessarily purely as drugs, but as procedures or interventions. I think about high-risk surgeries. I think about transplants that are incredibly complicated and involve multiple disciplines, from the anatomy of the surgery to the molecular biology of immune suppression to the management of the psychosocial management of the patient. I think of these as a realm where medicine has a lot to offer that is not traditionally thought of in a drug company paradigm. So my vision for this would have a multidisciplinary unit that's associated some centers of excellence where this type of care can be delivered, but with tools that are supported by industry, even if not under that model.
We call it genomic surgery then.
I think that that would be an apt term.
Could we have the question and the answer very short, please?
So I don't have any question actually, so I have 3 comments and I'll make it very short. And thanks, Tim, for the exciting talk. So we have been collaborating quite a bit in the N-of-1 collaborative, but also in 1M. So first comment, the regulatory situation in Europe is a bit different than in the US. So the ASO treatment is a hugely unregulated area. So we use the named patient framework for treating patients, which means that the time from identifying patients to treating patients is also unregulated. So we can actually shorten it quite a bit. And if we are really able to do it, we can do it in 12 months time or 18 months time. So depending on how quick we can do the treatment. Then second quick comment, if you have, so that's to all of you, if you have a patient which has a mutation, a splice switching mutation in the intronic region, please get in touch with us. So with 1M, because we need to check whether this patient is really treatable. It's not as easy as it seems. So it's quite difficult, at least for now, to identify respective patients. And yeah, I leave it at that. And thanks again for the very nice talk.
Thank you. Thank you.